You got Divya email? Reminder email?
Yes, so it's live on YouTube now. I'll begin at 12. Okay. So a very warm welcome to everyone who's joined us today to be a part of the Regional Center for Biotechnology's Science Setu lecture series. RCB is an institution of national importance established by the Department of Biotechnology, Government of India, under the auspices of UNESCO. The mandate of the center is to provide a platform for biotechnology education, training, and research at the interface of multiple disciplines. To celebrate India's 75 years of independence, RCB has organized the Science Setu lecture series, where our faculty will showcase their ongoing research activities. So it's my pleasure to introduce our first speaker of this series, Dr. C.V. Srikanth. Dr. Srikanth completed his PhD in 2005 at the CSIR Institute of Microbial Technology at Chandigarh. He then moved to the United States for a postdoc at Harvard Medical School and University of Massachusetts Medical School, following which he returned to India and joined RCB as faculty. His group at RCB works on the molecular biology of infectious and idiopathic inflammation, with the broad goal of understanding the key events that cause inflammation during infection and autoimmune disorders using the model bacterial pathogen Salmonella typhimurium. He is a recipient of the prestigious Wellcome DBT Intermediate Fellowship Award and has published his research work in journals of international repute, including Cell Reports, Nanoscale, and Journal of Cell Science. With that, I'd like to invite Dr. Srikanth to begin his lecture entitled The Interplay Between Salmonella and Host Epigenetics Machinery in Chronic Infections. Over to you, CV.
Thank you, Divya. Let me just share my slides. Yeah, so very good afternoon to all of you. Before I begin, I have to mention that it's actually an honor to be participating in this Science Setu lecture series, and being the first one, and it is itself a big deal for me. I thank the organizers, Divya, Deepti, and Nidhi, for giving me this opportunity. So as Divya mentioned, I'll be talking majorly about my work, ongoing work, dealing with Salmonella host pathogen interaction. And for today's lecture, I'll be talking on interplay between Salmonella and host epigenetics machinery in chronic infections. So the lecture will have majorly two parts. In the first part, I will describe a little bit of the background of Salmonella infections. And I would like to convince you that there are aspects of Salmonella infections which are rare and which are understudied. And then I'll move into the some of the recent data set that we have produced, which kind of which addresses those rare and understudied aspect of Salmonella disease. So the Salmonella pathogen, as it is, it basically most of the disease-causing entities of Salmonella come under two major groups: the typhoidal and the non-typhoidal Salmonellae. Now, these are all under the same genus Salmonella and the species Enterica. And then there are like thousands and thousands of different serovars and varieties, which are basically causing diseases in humans, animals, birds. And they can be kind of grouped under two major categories. One of them is the typhoidal strains, which cause a systemic form of illness. After ingestion, they would basically spread into the different parts of the body. They might settle down in spleen, liver, gallbladder, and then the disease sets in. The disease lasts for about 14 days. And in the recent times, typhoidal strain has been little low in incidence, but it hasn't still been eradicated. The other form, which is the non-typhoidal strain, they cause localized infections, which is going to be the main theme of my talk today. And together they are called as gastroenteritis, and it involves diarrhea, fever, abdominal cramps, and vomiting. Together, both the typhoidal and non-typhoidal strains, they cause significant economic burden, several deaths and hospitalization. Just to mention that each year, there are about 1.3 billion cases of non-typhoidal salmonella itself, and more than $2 billion are spent annually. And these are basically based on records that are there, and there are many, many cases, particularly in underdeveloped countries, where these diseases, and the money spent goes, unnoticed and unrecorded. And apart from this, there are also rare outcomes of the Salmonella disease, which is going to be the main theme of my talk today, wherein the bacteria actually, instead of getting resolved, which is usually the case in case of gastroenteritis, the bacteria is able to actually cause a persistent infections. Now, in the entire duration of my talk, I might be using certain aspect related to Salmonella typhimurium, which is a non-typhoidal strain of Salmonella disease. And you will see that some aspects of this is also kind of shared and is common between the typhoidal strains. Okay. So the way the Salmonella disease is caused is in this way, which is represented here in this particular graphics. After ingestion, somehow the bacteria gets through it thrives through the gastric juices and the low pH, and it would come in contact with the intestinal epithelial cells. So usually in the intestinal lumen is where the bacterial pathogenicity starts to take place. The bacteria would kind of come in contact with the intestine, and that's when it would start expressing several of its genes. And a major aspect that has to be mentioned here is this SPI1 and the SPI2 loci. These are the Salmonella pathogenicity island 1 and 2 loci, which are kind of, they are basically understandably horizontally acquired genome patches through which Salmonella actually causes its pathogenicity. Both these loci, they are able to actually encode for about 40 or so different proteins. Some of these proteins actually assemble and form a nano machine like apparatus as shown over here, and this nano machine would be able to actually inject the proteins which are of virulence importance into the host cell directly. So this machine is able to actually inject proteins that are made in the bacteria directly into the host cell, which are called as effector proteins. Now, this process would actually enable a pathogen like Salmonella to actually get into the epithelial cells, which are actually non-phagocytic. They are not supposed to actually uptake any bacteria, but because of these effector proteins, the pathogen is able to get into the host cell and then sit inside a vacuole, which is called as Salmonella containing vacuole. And from here on, the pathogen can either stay for a few hours and get killed in the host cell, or it can also stay for several days. And that's how the disease sets in. And while these things are happening, because of the presence of the effectors that the bacteria has produced and has kind of secreted, it actually elicits a very, very strong inflammation. And that's why this particular disease is also called as inflammatory diarrhea. Now, these effectors would actually attract the recruitment of several immune cells. And in this case, I have shown neutrophils over here, which would basically come from the basolateral aspect of the epithelial cells and come to the apical side. And there they would kind of try to induce their induce factors which would kill the bacteria, but also damage the epithelium and the tissue surrounding, which basically is the major burden. Okay, so by the SPI1 and SPI2 loci, the invasion takes place, pro-inflammatory responses caused by immune cell infiltration takes place. And at this point, the bacteria is also able to actually cause a large amount of transcriptomic reprogramming in the host. And by now, the bacteria has actually hijacked the host cell. Now, if you look at the bigger picture of this, bacteria has actually kind of come past the colonization resistance which would be offered by the commensal microflora, which usually prevent entry of any pathogen. It has come in contact with the epithelial cells by kind of passing through these thick mucus layer as well as antimicrobial peptide, which is usually secreted and is present in large amounts in the mucosa, and it has entered the epithelial cell, which is shown here in the zoomed image. Now, after that, once it has settled down in the Salmonella containing vacuole, the bacteria would also kind of either be eradicated or it would kind of enter immune cells which are just beneath the epithelial lining. And one of the first immune cells that would engulf a bacteria which has kind of come past the epithelium would be the macrophages and the dendritic cells. And that's where the bacteria lives most of its life when it is inside the host cell. Now, as I mentioned earlier, one of the themes of my talks would be the rare outcomes of non-typhoidal salmonella disease. And a major factor for these rare outcomes is actually the bacteria being able to settle down into a persistent form of infection. And that happens when the bacteria enters the macrophages, and then it can kind of utilize a dichotomy that exists in the macrophages, wherein the macrophages can either kill the bacteria by its pro-inflammatory mechanisms and antimicrobial activities, and those macrophages are usually called as the M1 type inflammatory macrophages. It is somehow known that in the persistent infections, the bacteria is able to actually convert these M1 type macrophages into the M2 type, which are actually non-bactericidal and anti-inflammatory. And thereby, inside those salmonella containing vacuole in the macrophages, similar to the epithelial cells here, the bacteria can actually thrive for long duration. In many cases, it has been seen that the bacteria can stay for more than a year. Now, when salmonella causes a persistent infection, there can be multiple outcomes. The first one is that it can lead to relapse of the disease. It leads to constant shedding of the bacteria through the fecal matter, which becomes a reason for further infections, passing on the infection to other individuals. And the third thing is that persistent infection causes persistent inflammatory mechanisms in the tissue wherever the bacteria is staying, the bacteria is residing. Usually these tissues are either from the gallbladder, liver, or the bone marrow, and that would lead to certain side effects such as gallbladder cancer, colitis. It can lead to colitis, or it can lead to colon cancer. And all these diseases that I'm mentioning right now, there are actually clear associations of salmonella infections. So they can be called as serious side effects of such persistent infections. Now, there are several genes that are involved when the salmonella converts the M1 macrophages to the M2 type, and it's like a massive reprogramming that occurs in the host cell in the macrophages. What has been known is that usually during the M2 switch, the macrophages go into a fatty acid metabolism type, wherein they are less inflammatory and they are less bactericidal. Now, the salmonella-mediated macrophage polarization, the mechanisms, etc., are not fully understood. Now, at this point, the take-home message would be that persistent infections in the distal organs may be key for tumorigenesis, because when they stay in these distal organs, such as gallbladder or in the colon, they can actually cause chronic inflammatory mechanisms, or mechanisms that would eventually trigger tumorigenesis. And I will try to convince you in the next few slides how and why this happens. So what is known at the moment is, at least for gallbladder cancer and its association with salmonella infection, is that once salmonella gets into the gallbladder, it basically causes MAPK and AKT dependent pathways, and these pathways can actually trigger tumorigenesis or pre-malignant changes, particularly in a scenario wherein proteins like c-MYC or TP53 are mutated. Now, this is basically a condition which can be called as a predisposition to cancer. So a host cell or an individual who has a predisposition for cancer, and he gets a salmonella infection, and if this bacteria gets into the persistent mode, or if it gets into these distal organs such as gallbladder or liver, That is where such tumorigenesis can take place. Okay, and what has been seen is that several unrelated studies, people have seen that marked epigenetic alterations take place, particularly in the gallbladder tissue when they have gallbladder cancer. A recent study which compared gallbladder cancer patients from United States and Chile, they actually found that there were marked epigenetic alterations. Now, what are these epigenetic changes? Epigenetic changes are basically heritable phenotypic changes without any change in the DNA sequence. Okay, so the bacteria is not actually altering the DNA sequence per se, but it is probably acting on the epigenetic machineries. So that is what was our original hypothesis when we started our study here. Now, particularly because while epigenetic alterations are known in gallbladder cancer, but whether Salmonella is doing it and what kind of epigenetic alterations are going on is not known. So we started our work with that type of a basis. So what we did was we did a very simple experiment wherein we infected Salmonella to an epithelial cell, which is HCT8 in this particular case. And we also had a control uninfected cell. And basically the total RNA from these cells were extracted, and they were basically subjected to a quantitative real-time PCR array. Now, this PCR array was majorly done to select or screen for epigenetic modifiers, if any, if they undergo any change in the expression alteration during Salmonella infection. So this is the list of the different genes that were used, and each of them involves different types of chromatin modifiers such as histone acetylases, histone deacetylases, histone methylases, demethylases, and so on and so forth. And what we found was something very interesting. We found that actually among the 50 different genes that were screened, most of them did not show any change or any alteration during Salmonella infection when we compared them with the control cell. But this one particular gene called as KDM6B, which is a demethylase, was seen to be significantly upregulated during the infection. Now, it was important to check whether this particular upregulation is actually taking place in just HCT8 cells or also in other cell types. So we used several different cells, HCT8 cells, CACO2, RAW264.7 macrophage cell line, or primary macrophages such as bone marrow derived macrophages. And we saw that in all of them, actually, when we infect with Salmonella, there was at least 5-fold or more upregulation of KDM6B. So that was basically the basis of our study, which kind of, we were able to conclude that KDM6B demethylase is upregulated during Salmonella infection. Now, it was important to check whether the KDM6B upregulation is actually linked to its function. So what is known is that when a gene is in a repressed state, there are certain types of proteins which are basically silencers that sit on that part of the chromatin and keeps that gene in an inactive form. But when that particular gene has to undergo activation, usually enzymes like KDM6B act on the histones and they remove the methylations on the histone H3K27, which is basically the lysine 27, which are usually trimethylated in the silent state. These trimethylations are reversed or removed by KDM6B so that that particular patch of the DNA can slowly become active and synthesis can take place. I mean, the gene synthesis can take place. Now, why this was important, and what I said earlier, that during the macrophage polarization, we know that massive gene activations and inactivation take place. So we thought that these type of KDM6B mediated methylations may have an important role for that. So we checked whether actually during Salmonella infection the methylation patterns change, particularly the H3K27 trimethylation. So control and infected cells were acid extract proteins were acid extracted so that we get the basic histone proteins from them, and then they were basically subjected to an immunoblot for particularly using antibodies for H3K27 trimethylation. Now, if you see the control cell and compare it with the Salmonella infected cell, we saw that the trimethylation was actually lowered, and when these cells were treated with a Salmonella surface protein, a surface molecule lipopolysaccharide, there was no change. A heat kill Salmonella also was unable to induce any change. And similarly, a mutant for the Salmonella pathogenicity island 1, which I mentioned earlier in the introduction, when that was used, we saw that there was actually no change in the S3K trimethylation. So that basically meant that the SPY1 loci was actually required for the S3K trimethylation changes that happens during Salmonella infection. Now, to check this by another experiment, we also used two important regulatory components, a HIL-A and a HIL-D, which is required for the SPY1 protein, effector protein secretion. And in both the cases, we saw that the S3K trimethylation was kind of not altered as we see it in the wild-type Salmonella case. So we concluded that the KDM6B upregulation is accompanied with a decrease in S3K trimethylation mark, and the recruitment of the KDM6B requires a live Salmonella with a functional SPY1 loci. Now, it was important to see where all in the genome the KDM6B actually is recruited to. So for that, we actually did a chromatin immunoprecipitation clubbed with an RT-PCR array, wherein control and Salmonella infected cells were actually subjected to chromatin immunoprecipitation, wherein we first break the chromatin into small 200 to 500 base pair long molecules, and then they are basically preferentially immunoprecipitated using a KDM6B chip grade antibody, so that you isolate the part of the genome wherein KDM6B is bound. And then this particular DNA is subjected to quantitative real-time PCR to identify where all it has bound to. We particularly used a signaling-based array, which was linked, which was basically having genes related to the various signaling pathways that may be required for the macrophage polarization. And what we see is that there were actually several different proteins, regulatory proteins, that were altered. Some of them are actually mentioned over here. And for my today's talk, I'll kind of focus on Ppar delta, which is a protein known to be involved in macrophage polarization, which was kind of done by some earlier studies, but unrelated to gallbladder or Salmonella infections. So first of all, when we checked the actual expression of Ppar delta during Salmonella infection, we saw that at both 4 hours and 18 hours post-infection, Ppar delta actually got upregulated during infection. Okay, so we kind of concluded that KDM6B is recruited to promoters of several regulatory genes from our chip assay. And we saw that Ppar delta is one of the KDM6B target gene that is seen to be upregulated during Salmonella infection. Now, it was important to further see how exactly KDM6B is modulating Ppar delta expression and whether it is actually a KDM6B activity or not. So we actually chose the Ppar delta promoter, and we kind of looked at the S3K trimethylation based chromatin immunoprecipitation, and we saw that particularly in the Ppar delta promoter during Salmonella infection, which is shown in this bar, actually it goes down. And so the trimethylation of Ppar delta promoter is going down during Salmonella infection, whereas in a housekeeping gene such as GAPDH, we don't see any difference. Not only this, when we treat the cells with a RNA interference specifically for KDM6B, so if you treat cells with an siRNA to downregulate or knock down KDM6B during infection, we don't see any change in the Ppar delta expression as compared to the control siRNA treated cells. So we concluded that Ppar delta gene promoter is remodeled by KDM6B, and KDM6B demethylase activity is required for Ppar delta activation during infection. So now I'll come back to the original quest of the macrophage polarization. So if you remember, I had mentioned that Salmonella remodel host macrophages to the M2 type, particularly during chronic infections. And what is known till now is that one of the Salmonella effectors, STEE is required for the reprogramming of the macrophages from M1 to M2. And now it was important to check whether KDM6B has a role in macrophage polarization. So that's what we checked in the next experiment. So for this, now we actually used the raw 264.7 macrophages, and when they were infected with Salmonella, and they were also infected to a raw macrophage which was actually knocked down for the KDM6B, we saw that at 4 hours post-infection, there was no change in the actual colony-forming units of Salmonella in the macrophages. But at 18 hours, compared to the control cells in the Kadium-6B saRNA treated cells or the knockdown cells, we saw that Salmonella colony forming units were substantially lowered. This meant that Kadium-6B had a role in macrophage biology during Salmonella infection. And next, what we did was we actually looked for the fold change of Kadium-6B, both in a wild-type Salmonella and a STE-treated Salmonella, which basically was one of the M2 macrophage polarization, mediating effector. Now, in both the cases, we did not see in RT-PCR as well as in Western blots, we did not see any change. We did not see any change in the STK trimethylation also in a wild type as well as a STE mutant. So that meant that this particular pathway of macrophage polarization was not STE dependent. It was a SPY1 dependent mechanism, which kind of occurs through epigenetic mode. So we hypothesized that Salmonella-mediated M2 macrophage polarization may depend on Kadium-6B epigenetic activity. So now we kind of looked further into by using an in vivo model of chronic infection. So for an in vivo model of chronic infection, usually an engineered Salmonella strain, which is a ROA deficient strain, is used. So ROA is a aromatic amino acid deficient Salmonella, which when infected to mouse would kind of continue to thrive in the animals, and it is also known that these particular bacteria go into distal organs. Now, if you do similar experiments with a wild type bacteria, it would kill the animal in about 7 days post-infection. So for a chronic setting, this type of a mutant ROA bacteria was required. So post-infection, we kind of had one group which was kind of which was untreated. There was no other treatment apart from Salmonella infection. But the second group we actually treated with a specific drug for Kadium-6B, which is called as GSK-J4. So this drug is known to be a specific inhibitor for both Kadium-6B and Kadium-6A. And we have already seen in an earlier experiment that Kadium-6A had no role in our studies. Now, post-infection, at around 30 days, when the animals were euthanized and the different organs were tested, at the moment I'm just showing here the data from spleen, we saw that the colony forming units in the vehicle treated animal was very high, but in the GSK-J4, which is basically the Kadium-6B inhibitor, was actually very, very low. So this meant that if you inhibit Kadium-6B, both in vitro and in vivo, it would actually inhibit Salmonella's capacity to invade and colonize in the host. Now, to see whether this particular disability had anything to do with the macrophages, what we did was we used the flow cytometry, and we looked at specifically the using antibodies for macrophages. Here, the specific one shown for the M2 macrophages, the CD206 as well as the CD301. What we saw is that compared to control animals, the Salmonella infected animals had a higher amount of M2 macrophages. But when we see the same trend in the when we see the same feature in the GSK-J4 treated animals, we saw that the GSK-J4 animal had actually a substantially decreased amount of M2 macrophage conversion. So we were able to conclude that inhibition of Kadium-6B renders lesser bacterial burden at 30-day post-infection in chronic infection model, and the M2 macrophage polarization increase that we see during Salmonella infection is blocked by GSK-J4 treatment. So that means the M2 macrophage polarization dependent on Kadium-6B. Now, it was important to see whether these macrophages that we see were basically behaving differently by when we inhibit the Kadium-6B were actually M2 macrophages. So we checked for individual macrophage M2 markers such as the arginase, the IL-10, and the YM1. So the arginase is actually it's an enzyme that converts ornithine. It converts arginine to ornithine in the cycle, so that the fatty acid metabolism change can take place. The IL-10 is an anti-inflammatory cytokine, and YM1 is also it's a gene that is required for the fatty acid metabolism. So usually it is known that during macrophage M1 to M2 shift, they actually undergo a metabolic change from the regular energy and metabolism to a fatty acid metabolic cycle. And we saw exactly that in Salmonella infection, but this was prevented, as you can see with these bars, when we actually use a GSK-J4 drug, which was specific for KDM6B. We also looked at several other targets of KDM6B, and we saw that they all were actually differentially behaving when we either treat this host with GSK-J4, or if the KDM6B was knocked down in the host. So with all this, we concluded that inhibition of KDM6B activity results in blockade of macrophage polarization. Okay, so that will basically conclude a major part of my talk, and I will now slowly move to the second aspect, wherein I had mentioned earlier that this type of persistent infection as well as macrophage polarization may have an important role to play in carcinogenesis, particularly in gallbladder cancer. So for that, we first of all checked whether in these type of infections that we do, in the chronic infection, what is the status of KDM6B in the gallbladder. So first of all, we saw that in chronic infections in the gallbladder, we were able to see that the KDM6B was actually upregulated in the animals. And we also saw that the gallbladder of a regularly infected mice had significant amount of Salmonella, but if these animals were treated with GSK-J4 drug, the bacterial burden in their gallbladder was actually substantially reduced. So we concluded that KDM6B is recruited in murine gallbladder in chronic Salmonella infection, and inhibition of KDM6B activation results in a compromised infection. So to see whether the gallbladder carcinogenesis and KDM6B or the Salmonella infection has a direct role, we developed a murine model, which basically was used by feeding the animals with a lithogenic diet so that the gallbladders develop gallstones. Now, gallstones and Salmonella infection both have been associated with gallbladder cancer. So our hunch was that if we kind of do both, if we produce gallstones in these mice, and if those animals are actually infected with Salmonella with a chronic infection strain, we might get a hint about what exactly is happening. So post-infection and post-euthanization, we saw that compared to the control animals, the lithogenic diet-fed animals had enlarged gallbladders, and those gallbladders actually had gallstones, as you can see over here. And when we looked at the specific markers that we had earlier identified in our study, so to name them like the KDM6B, we saw that it was actually upregulated in the gallbladders of these animals. And we also saw that the Ppar delta was actually upregulated in these animals. And we also saw the HDAC, which is another epigenetic modifier, which was seen in the US and Chile gallbladder cancer patients, was actually going up. And so with that, I will actually conclude here. But just one last piece of data. When we looked at the histopathology of these animals, we saw that compared to the control animals, the lithogenic diet-fed animals and the Salmonella infection case, we saw there were actually several changes in the gallbladder. We saw that there was actually clear-cut signs of inflammation, which is kind of represented by these red arrows in these zoomed in images you can see. And we also saw signs of metaplasia as well as epithelial erosion, and we saw signs of high degree of cell division. Now this was actually 4 months post-infection. If you look at the 8.5 months post-infection, we also saw the same type of features, and they were actually much more pronounced, as you can see over here. There was actually metaplasia and other signs of pre-malignant lesions. So now these are actually wild type animals. If similar infections are actually done on animals that are, let us say, predisposed to any type of cancer, such as with a semi-mutation for c-MYC or APC, we expect that this might actually lead to clear-cut tumorigenesis. Now, with that, I will conclude here that Salmonella infection actually results in induction of pre-malignant lesions. What I have conveyed today is that during Salmonella infection, we see an upregulation of KDM6B, which is a histone demethylase. And this demethylase activity actually occurs in several genes in the genome. And one of the major entities was the Ppar delta, whose promoter undergoes remodeling by KDM6B that leads to fatty acid metabolism gene activation, and which converts the macrophages from M1 to M2 type and allows the bacteria to settle down into chronic infections. And during these times, the other changes that take place in the host are actually an upregulation of HDAC7, CBX4, about which I didn't talk about today, and RUNX2 and several other genes, which leads to pre-malignant changes. Now, with that, I will kind of end my talk here, and I would like to definitely acknowledge the people who have kind of done a lot of work in this. Most of the work that I presented was done by a former PhD student, Sarika Rana, who has now passed out and gone for her postdoc. She's working in Belgium. The last bit of part was actually done by a senior PhD student, Sonalika. I have to thank the other members of the lab. I also would like to thank the executive director of RCB Instrumentation Facility, the parent body, Department of Biotechnology, my collaborators, Dr. Sujoy Paul, Dr. Pallavi Shetripal, Dr. Pramod Garg, Dr. Vinita Huja, and Girish Ratnaparke. The major funding for this was actually from DST SERB. The other funding source that I have is STARS MHRD and DBT Northeast Grant. And with that, I will end here, and I will be happy to take questions if there are any. Thank you all.
Thank you for that very interesting talk, CV. So if anybody has questions, you can either type it in the chat box or raise your hand, and I can allow you to ask the question. So we do have a couple of questions already in the chat box, CV. So one is from Harsh Oza. His question is, are these epigenetic changes caused by Salmonella to promote its survival, or does it happen as a host defense mechanism?
Okay, so based on our data, what we see is that if we block the KDM6B upregulation, we see that actually the bacterial burden both in vitro and in vivo it goes down. So that most likely, it kind of points towards the thing that this mechanism is being activated by the bacteria for its own benefit. As I said that, the bacteria would like to convert the macrophages, for example, the macrophage environment from M1 to M2 type for its survival, because the M1 is antibacterial, it has bactericidal activity. The bacteria would like to convert it into an M2 type for a chronic carriage. So it's definitely for the sake of bacteria's own growth.
Okay.
And whatever else that we see is, I would say, the side effect of this.
And there's a question from Sakshi Khera. So she's asking how M1 to M2 polarization affects the fatty acid metabolism. And does this change in metabolism promote the persistent infection of bacteria in these macrophages?
Yes. So basically during the M1 to M2 conversion, as I said, huge amount of gene expression alteration take place. So the entire macrophage physiology is kind of changed. So both the transcript at the transcriptome and the signaling level, the macrophage actually undergoes a rewiring. And this rewiring, what we think is happening due to the major epigenetic change that we pointed out. Now the metabolic shift from the regular to the fatty acid metabolism is majorly because I think for this type of a change that the macrophage has to undergo in a short period of time, it has to actually change its energy source. It has to change its metabolic profile so that it is able to take this load of energy flux, so that the other genes get activated, which are required for the anti-inflammatory and pro-bacteria setting.
Okay.
Yeah.
There is another question from Harsh again. Are there any epigenetic drugs known which can revert these epigenetic changes and make the macrophages more bactericidal?
Okay, so I think the clear-cut answer for this would be the GSK-J4 drug, which kind of directly acts on both the KDM6A and 6B. And we know that KDM6A has kind of no role in Salmonella infection. As I didn't show that data, but we did not see any change in KDM6A in any of our experiments. Now I have to also tell that this particular drug GSK-J4 has been used for acute myeloid leukemia for a similar type of a approach of changing the epigenetic machinery so that the host cell is relieved. And if I'm not very sure, but I think it is already in clinical trials.
Okay. Thank you for those answers. Are there any other questions? No, please. Okay, there's one more. This is from Shaminder. What is the concentration of the GSK drug that resulted in almost 50% reduction in the CFU per gram?
The drug that we give to the animals is intraperitoneal, and we give about 5.6 milligrams per kilogram of the mice. Yeah. So in all the experiments, we had used that concentration only. But otherwise, it has an IC50 of about 60 nanomolar, this drug.
Okay. I don't currently see any more questions. So I'd like to thank you once again, CV, for the wonderful presentation. And thank you all for attending this.
Thanks, CV, for agreeing to give this talk.
Yes.
Thank you, all of you.
Okay. We can now end this meeting.